Check it out now, y'all.
NanoHub U, online instruction.
Welcome back. Today we'll be talking about the homework solution. You know that we have covered a lot of material in this segment that we have discussed, for example, various types of selectivity issues, in particular 3 types of selectivity, right? Incorrect binding, for example, and then there was this random sequential absorption where the molecules, parasitic molecules, were sort of landing in random spaces and not really binding to the target receptor molecules, but sort of parasitically getting attached to the sensor surface. And that created parasitic signals, false positives that we didn't want. And of course, there was this noise issue that we discussed. So this in this homework, we'll be talking about a few problems on the selectivity. And then there were 2 or 3 lectures on genome sequencing. I will run a few things by you. So once you have done this homework, if you have done it carefully, you should have a very deep understanding about various processes involved in selectivity issues. So the first problem 5.1, had to do with the energetics of binding. The bottom line, you understand the problem, right? The bottom line was that if 2 DNA were perfectly conjugate, well, they should bind very strongly, and that is what we want. But it turns out many cases, even if there is a parasitic molecule which should not have bound ideally, still binds and gives a false parasitic signal. So this is what we're trying to understand, that how strong are those? Can we increase the temperature of the solution a little bit so that all the parasitic molecules that got bound can be melted away and sort of taken away, washed away? So that's what we're trying to understand. So let's see the difference in binding energy between matched and mismatched DNA. So we just gave a specific sequence. You're supposed to use a particular tool which calculates the corresponding binding energy. And then essentially you were asked to calculate the corresponding energy. Now this tool actually implements the equations, the small analytical equations that I discussed in the class. And so therefore, although this tool looks like a black box, actually the equations were already discussed in terms of the melting temperature that you may remember from the lectures. So therefore, you shouldn't really be afraid to ask questions about these results of these using this simulation software. So let me show you some example. For example, I gave you 2 sequences. The first sequence is, this is the receptor molecule, you see, starting from 5 prime. Then you have a 22 base pair in here, ending with 3 prime. What does it mean? First of all, this is easy, right? This is A, T, C, G. So I have a particular sequence. This is a, let's say, potentiometric sensor surface. I have a 22 molecules sort of attached and standing straight like that on the sensor surface. So this is the 22. Now what is this 5 prime? Well, if you look at a DNA molecule, and I just copied it from the web, that you will see one end of it. It's inside everything is nicely matched, but there are 5 carbon molecules, 1, 2, 3, 4, 1, 2, 3, 4, and 5. And these 5 molecules on the carbon molecules on one end, and then correspondingly in the 5th location there is next phosphate group. And similarly on the other end, on the 3rd position, the next phosphate group will attach. So bottom line is, there's this 5 prime and 3 prime simply says the orientation of the molecule. That's this 5 prime and 3 prime. And what we asked, whether the binding energy associated with this, with another one. And this is the bottom one, is the target molecule. Now you may be thinking that this doesn't look very complementary. After all, there is this T prime. T cannot possibly conjugate with a T. But you see, the way you have to think about it, this 5' on the top always goes with 3'. So you should be looking from this side onward. The 5' side should be going from this side onward, and now you can see it's complementary because A goes with T, G goes with C, T goes with A, and so therefore if you just sort of flip it, you will see the secondary sequence or the target sequence is exactly complementary. This is how it is also. 5', 3' on this side, and 5' goes with the 3', and then correspondingly these two chains sort of line up. So we'll be thinking about how strong this binding is. And this is the mismatched pair. So for example, this is the same sequence as the first one, because remember this top one is the receptor, right? And then this bottom one is the mismatched DNA. That is, this is the parasitic DNA. Once again, you can check very quickly that if you start from 3' and match it with 5', then you will see that there is a T going with this T, which will not bind. And therefore, you can immediately see as you walk down this sort of set of letters that is not going to bind very strongly with the primary sequence, right? So we want to know what is the difference between the target, which is this sequence, versus the parasitic sequence, which is the false signal, false positive signal. And we want to see how strong are what is the difference in the binding energy? Can we selectively bind with each other? So that's the question. So let's see. So once you have sort of put these sequences in the simulator and run it, you'll get some numbers. And the numbers, oh, by the way, I just wanted to emphasize that the goal is that if the binding is perfect, then there'll be very strong bonds between the receptor and the target. And with the imperfect binding, there'll be many missing bonds. And then therefore, this would be a weaker bond. And so in a droplet, if you increase the temperature by a transistor, let's say, these ones will debond and will be washed away. Only the proper signal will remain. That's how we enhance selectivity, right? So let's see how the two sequences that we talked about, how strong are the binding. So there are, this you have to look at a little bit carefully. So for example, once you simulate the structure, you will get a whole set of numbers. And what I want to walk you through the results a little bit. Let's first read this first line. It says delta G is 35.09 kilocalorie per mole. You know, mole is there's Avogadro's number of particles. So it is saying that this is the total binding energy of that many molecules. So first of all, you know that 1 kilocalorie is 4.2 kilojoules, and 100 kilojoules per mole is approximately 1 eV per bond, per biomolecule, and 1 eV is 40 kT. So therefore, you could immediately convert this number by just using these three sets into the binding energy in terms of kT. You can do that. Now take a look. Remember that 5' goes with the 3', and you can see that all 22 bonds are now completely bound together. And this solid line says that these are strong bonds, and the bonds, consecutive bonds, are all essentially has been aligned. They are properly bounded. So it's very strong, 35.09 kilocalorie for perfect matching. But the interesting thing is that even when things are perfectly matched, there may be many cases where they will not line up properly. They will bind, but they bind very weakly. For example, take a look at 2. Remember, this whole page is for perfect binding. But perfect binding is not, although the molecules themselves can in principle bind properly, which is this case, there will be many cases where they don't bind properly, even though in principle they could. Look at this too. In this case, delta G is this minuscule 1.95 kilocalorie per mole, and only 2 base pairs have bound properly. They could in principle bind in 22 places. But statistically, when the molecules are coming in contact with each other, they can incorrectly bind where rest 22 are not bound. For example, take a look. What has happened that the bottom molecule, the target molecule, is short of come and gotten shifted. So there is 2 places, C going with G, A going with T. The neighbors are bound. There are few other places given by these double dots, 4 other places where they are bound but their neighbors are not. So relatively very weak bonds. And in that case, you can see there's only 2 base pairs that have bound strongly, neighboring base pairs, and the binding energy is very, very small. And you would say there are whole sorts of combinations. So therefore 2, 2 in here, 2 for various types of combination going through the whole stack. And in each case, you have about 1 or 2 kilojoules, kilocalories per mole, very weak bonds. So although in this case, we have perfect binding, you see, but in principle, you will see you'll have a lot of cases where statistically the binding is not so strong. So you can count 1, 2, 3, 4, 5, 6, 12, 14, 15, 16 said there is about 18 in here. 1 in 18 cases, the binding will be very good. This is the challenge, that even when the sequences are complementary, it is difficult to make sure that they get aligned properly and they are bound strongly. There are a few cases, in fact 2 cases, where you have 3 binding. Instead of 22, you have 3 binding. So we have 22 binding, 3 binding, 2 cases, and 2 binding the remaining cases. So only one of them is what we are really after. Okay. Now this is for the case of perfect match. What about imperfect match? Well, for the case of imperfect match, again you can go, remember that's the second sequence with the parasitic molecule. This time things are a little bit better, because you can see that in the imperfect match, although it's imperfect, but there are at least 5 places where the neighbors still find within the long sequence there is good binding. There are a set of about 6 where the binding is relatively weak. And on average, you have for the 5, you have about 6.71 kilocalorie per mole, approximately 1 or slightly more than 1 kilocalorie per mole. And then there are a whole set of places where 3 binding is possible, 2 bindings are possible. You can see these 2 vertical lines, and all these are 2 bindings. And sometimes you may have this. So this large number of 2 bindings, 1 case of 3 binding, and 1 case of 5 binding. So tell me this, what will be the difference in the selectivity? In the previous case, the best was 22. Right? In here also the best is about 5. So I have to make sure that I raise the temperature of the solution after all the bindings are done enough so that at least these 5 bindings cases are detached. Of course, if I can make sure that 5 binding cases are detached, that means in the previous slide all these ones will also get detached. All the 3 ones will also get detached. Only thing that will remain is the 22. So therefore, this type of calculations are very helpful, you see, so that you can decide ahead of time that what type of temperature do you need in order to have high selectivity. Okay. Now the next problem we'll be talking about has to do with this space-based selectivity problem. You remember that the molecules can random places not covered by the receptor, and you can have a selectivity problem. So so this is already specified, the problem is specified, the solution is given, but let me show you what we are really after here. So this is a this is a sensor surface. We are looking from the top. And what you see here in this blue is initially these were all sort of the receptors. The empty blue circles were receptors sitting there on their own. How many receptors? You can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So there were 10 receptors within this sensor surface waiting for the target biomolecules to come. And you incubate it for a certain period of time. The target biomolecules has indeed come, and it has occupied 1, 2, 3, 4, 5, 6, 7, 8. 8 out of the 10 is now occupied. And what would happen that the rest will be occupied by, in these places, this red are the parasitic molecules. The parasitic molecules, of course, would not fit with the receptor itself, so they are sitting in empty places. So let's first say how many solid blue or the target molecule have we captured. So that will be given by theta divided by pi r squared. Theta is the fraction per centimeter square, the fraction of the area covered by the target molecule, and divided by pi r naught square. R naught is the radius of this target. Now, the remaining place is now available for the biomolecules to sit. And in that case, it will be theta infinity. What is theta infinity? Theta infinity is that when the entire surface is covered, then the fraction of area that will be covered by this molecule, and in this case, you know that answer is 0.54. Theta infinity is about 0.54, but of that 0.54, blue has already taken, the theta has already taken a certain amount of space. The remaining space can be filled by the parasitic molecule, pi r squared. So that gives me what is the potential number of parasitic molecule that can sit here. All right, so it's very easy then. So what you need to do is, for example, in theta here, for theta, you substitute this quantity in here, solve for it, and it gives you number of molecules, blue molecules that were captured, divided by number of parasitic molecules that have been captured. And of course, this is related to signal to noise ratio. And the signal to noise ratio, of course, this is the charge of each target molecule. Let's say each DNA carries 10 charge. So sigma t would be 10, for example. And sigma p is a charge carried by the parasitic molecule. Often it may carry a small amount of charge. Let's say 1. So sigma p would be 1. Kt is the rate at which things bind, and Kp is the rate at which the red molecules bind. They come and sit on the sensor surface. And then, of course, there is this initial density of the target molecules. The higher the density, the more easier it is for the molecules to sort of settle on the sensor surface. And N naught divided by Np. That's the N naught over Np. So if you do this, substitute this quantity, then you can immediately solve for this value N naught, and then N naught, you see everything inside here is known. So for example, we want a signal to noise ratio of 100. So SNR of 100 will go here, the charges will go here, the KT and KP, these things are known, and rho T is already given, target density of target molecule, rho P is the density of the parasitic molecules, these are all given, radius unknown, and so you can calculate N naught. And the N naught value you calculate in this case is 5.6, 10 to the power 10 centimeter per centimeter square. So N naught is essentially the number of places where the receptors can essentially come and sit. Okay. And if you do the corresponding simulation, you will see that you would get about the same number. You can see 5 times This was done by this analytical calculation, 5.6 10 to the power 10. And in this case, you see for a signal to noise ratio about 100, you get about the same number. And the important thing is that the higher the number, better is our ability to selectively bind to this surface. So signal to noise ratio would be much better. Now, one thing I want to emphasize that this N naught essentially involves all this red plus this blue together, right? And so in some places, only NP is coming in, and in some other places, the target molecules are coming in. So the theta is the fraction that is taken up by the target molecule, and the red ones are the places that were taken up by the parasitic molecule. Okay. Now, it is always better that if you had molecules which are parasitic molecules which are bigger size, because if it has a bigger size, then it will not be able to sort of bind as easily. And so that is having this parasitic molecule which has a bigger size enhances the signal to noise ratio, and that is given by the next problem that you may have solved for this selectivity problem. This is, of course, using the biosensor lab tool.